Welcome to another video of Unit 3. Let's start. Blood sugar regulation in the body. Blood sugar, glucose, regulation is a complex process that involves various hormones and metabolic pathways to maintain a stable blood glucose level, crucial for proper body function. Hormonal regulation. 1. Insulin. Source: pancreas, beta cells in the islets of Langerhans. Function: lowers blood glucose levels by facilitating the uptake of glucose into cells for energy production or storage as glycogen in the liver and muscles. 2. Glucagon. Source: pancreas, alpha cells in the islets of Langerhans. Function: raises blood glucose levels by stimulating the liver to break down glycogen into glucose, glycogenolysis, and produce glucose from non-carbohydrate sources, gluconeogenesis. 3. Cortisol. Source: adrenal glands. Function: stimulates gluconeogenesis, the process by which the liver generates glucose from non-carbohydrate precursors. It also makes cells less sensitive to insulin, raising blood glucose levels. 4. Epinephrine, adrenaline. Source: adrenal glands. Function: stimulates the liver and muscles to release glucose by promoting glycogenolysis. It also inhibits insulin release and increases blood glucose levels during stress, fight or flight response. 5. Growth hormone. Source: pituitary gland. Function: stimulates glucose production in the liver and reduces the uptake of glucose by cells, raising blood glucose levels. Diabetes mellitus. Definition: diabetes mellitus is a group of metabolic disorders characterized by high blood glucose levels due to the body's inability to produce sufficient insulin or respond to insulin properly. Classification. 1. Type 1 diabetes. Cause: autoimmune destruction of insulin-producing beta cells in the pancreas, leading to little or no insulin production. Management: insulin therapy is required for blood glucose control. 2. Type 2 diabetes. Cause: insulin resistance, cells do not respond effectively to insulin, and eventual beta cell dysfunction. Management: lifestyle changes, diet, exercise, oral medications, such as metformin, and in some cases, insulin therapy. 3. Gestational diabetes. Cause: occurs during pregnancy when the body cannot produce enough insulin to meet the demands of pregnancy. Management: often controlled with diet and exercise, but sometimes requires insulin therapy. 4. Other specific types. Result from genetic mutations or secondary to other diseases or conditions, example pancreatic diseases, hormonal disorders. Abnormalities of metabolism of carbohydrates. Hypoglycemia, low blood glucose levels, often due to excessive insulin or insufficient food intake. It can cause symptoms like shakiness, confusion, and in severe cases, coma. Hyperglycemia, high blood glucose levels, typically seen in untreated diabetes, leading to long-term complications such as cardiovascular disease, nerve damage, kidney failure, and vision problems. Metabolism of biomolecules. 1. Carbohydrates. Digestion and absorption. Carbohydrates, example starch, are broken down into simple sugars like glucose in the digestive system. Glycolysis, glucose is converted into pyruvate in the cytoplasm, releasing energy, ATP. Gluconeogenesis, the liver synthesizes glucose from non-carbohydrate sources, example lactate, amino acids. Glycogen synthesis and breakdown, excess glucose is stored as glycogen in the liver and muscles, glycogen is broken down to glucose when needed. 2. Proteins. Digestion, proteins are broken down into amino acids in the digestive system. Amino acid metabolism, amino acids are used for protein synthesis or converted to glucose, via gluconeogenesis, or other metabolic intermediates for energy. 3. Lipids. Digestion, fats are broken down into fatty acids and glycerol. Beta oxidation, fatty acids are converted into acetyl-CoA, which enters the citric acid cycle to produce ATP. Lipogenesis, excess glucose and amino acids can be converted into fatty acids for storage. 4. Nucleic acids. Digestion, nucleic acids, DNA/RNA, are broken down into nucleotides. Metabolism, nucleotides are used to synthesize new nucleic acids or can be metabolized for energy. The metabolic pathways of carbohydrates, proteins, and lipids are interconnected to maintain energy balance in the body. Dysfunction in any of these pathways can lead to metabolic disorders like diabetes. 1. Which enzyme is responsible for the conversion of glucose to glucose 6-phosphate in the liver? A. Hexokinase. B. Glucokinase. C. Phosphofructokinase. D. Glucose 6-phosphatase. Answer, B. 2. In type 2 diabetes, what is the primary defect that leads to hyperglycemia? A. Decreased insulin secretion. B. Insulin resistance. C. Overproduction of glucagon. D. Increased glucose absorption in the intestines. Answer, B. 3. Which of the following hormones is most involved in increasing blood glucose during fasting? A. Insulin. B. Glucagon. C. Cortisol. D. Epinephrine. Answer, B. 4. Which process in the liver results in the conversion of non-carbohydrate precursors, such as lactate, into glucose? A. Glycogenolysis. B. Glycolysis. C. Gluconeogenesis. D. Pentose phosphate pathway. Answer, C. 5. Which of the following is a primary characteristic of type 1 diabetes? A. Insulin resistance. B. Autoimmune destruction of pancreatic beta cells. C. Reduced sensitivity to cortisol. D. Increased insulin production. Answer, B. 6. The presence of which substance in urine is most indicative of uncontrolled diabetes? A. Protein. B. Glucose. C. Ketones. D. Bilirubin. Answer, B. 7. Which of the following is not a complication associated with chronic hyperglycemia in diabetes? A. Diabetic retinopathy. B. Diabetic nephropathy. C. Increased bone density. D. Cardiovascular disease. Answer, C. 8. Which of the following molecules directly inhibits glycogen synthase in the liver? A. AMP. B. Insulin. C. Glucagon. D. Glucose 6-phosphate. Answer, C. 9. Which of the following processes is directly stimulated by insulin in muscle tissue? A. Lipolysis. B. Glycogen synthesis. C. Gluconeogenesis. D. Glycogenolysis. Answer, B. 10. In which part of the cell does glycolysis occur? A. Mitochondria. B. Nucleus. C. Cytoplasm. D. Endoplasmic reticulum. Answer, C. 11. Which of the following best describes the effect of cortisol on glucose metabolism? A. Increases glucose uptake in peripheral tissues. B. Increases insulin sensitivity. C. Stimulates gluconeogenesis. D. Decreases liver glucose production. Answer, C. 12. Which enzyme is responsible for converting glycogen to glucose 1-phosphate during glycogenolysis? A. Glycogen synthase. B. Glucose 6-phosphatase. C. Glycogen phosphorylase. D. Phosphoglucomutase. Answer, C. 13. Which of the following molecules is the most immediate energy source for cells during glycolysis? A. ATP. B. NADH. C. Glucose. D. Pyruvate. Answer, C. 14. In diabetes, prolonged hyperglycemia may lead to the accumulation of which of the following compounds in the body? A. Acetoacetate. B. Fructose 6-phosphate. C. Glucose 1-phosphate. D. 3-hydroxybutyrate. Answer: A. 15. Which of the following molecules is produced during the pentose phosphate pathway and is used in nucleotide synthesis? A. NADH. B. NADF. C. FADH2. D. Acetyl-CoA. Answer: B. 16. Which of the following best describes the metabolic effect of epinephrine during stress? A. Decreases blood glucose. B. Increases insulin secretion. C. Stimulates glycogen breakdown and glucose release. D. Inhibits lipolysis. Answer: C. 17. Which of the following is a key characteristic of gestational diabetes? A. It is caused by insulin resistance during pregnancy. B. It always leads to type 2 diabetes. C. It involves an autoimmune attack on pancreatic beta cells. D. It typically resolves after childbirth without treatment. Answer: A. 18. Which enzyme is responsible for the irreversible conversion of pyruvate to acetyl-CoA in mitochondria? A. Pyruvate dehydrogenase. B. Citrate synthase. C. Malate dehydrogenase. D. Phosphenol pyruvate carboxygenase. Answer: A. 19. Which metabolic pathway is responsible for the synthesis of glucose from lactate? A. Glycolysis. B. Glycogenolysis. C. Gluconeogenesis. D. Lipogenesis. Answer: C. 20. What is the role of GLUT4 transporters in muscle and adipose tissue? A. To transport glucose into cells in response to insulin. B. To transport glucose out of cells during fasting. C. To convert glucose to glycogen. D. To regulate insulin secretion from the pancreas. Answer: A. Thanks for watching.